Dr. Brock is professor of surgery and oncology, as well as environmental health sciences at Johns Hopkins University. He's received multiple honors, including a Lifetime Achievement Award from the Bermuda Healthcare Foundation and the prestigious James IV Association of Surgeons Award, which included visiting professorships to medical schools in China, Japan, France, and South Africa. Dr. Brock is a member of numerous national medical societies in the US, most recently served as the president of the Society of Black Academic Surgeons. We had the news recently that Dr. Brock was named as first Dr. E.F. Gordon Endowed Professorship. We understand that this endowed professorship will allow him to continue his research on such things as developing novel molecular biomarkers to diagnose cancer much earlier. Today, Dr. Brock will examine the topic of COVID-19 vaccinations. He will speak to the fact that many persons are still reluctant to receive the vaccinations. Dr. Brock.
Well, thank you very much, Mr. Richardson. Really appreciate that. And I really appreciate the invitation to come and speak to you, albeit from afar. You know, it's hard to believe that it's been 40 years since I first spoke at Rotary after Sands Rotary, and I understand there are some Rotarians from Sands in the room who sponsored me to go to Japan, and I'll always be really deeply grateful for that opportunity. It's been so long since I've talked to you, I asked my dad, you know, what is the absolute longest time I can speak to you? And he said 25 minutes. So I'm going to take the prerogative and do a 25-minute debrief because I have an ambitious task. I really want to give you enough scientific concepts about the COVID vaccine to enable you to recognize this disinformation when you see it and make some sense of least of what's happening in all this internet chatter that I understand is going on in Bermuda as well. So first I would like to say that, can you see my screen pretty well?
Yes. Yes.
Okay. I want to say that my disclaimer is that I've had both of my shots. They both were Pfizer, and Bermuda has Pfizer. So we're going to talk a lot about the Pfizer vaccine today. So I'm going to first start with a brief history of vaccines. Then we're going to talk a little about DNA, RNA, and protein, what they are and how they work. And then vaccine efficacy or effectiveness and safety. And then we're going to touch a little bit on this hesitancy and objections and myths scenario. So in the latter half of the 1700s, it was well known that milkmaids never seemed to really get sick with smallpox, especially if they were milking cows that had cowpox, which is a milder version of smallpox. But it was Edward Jenner who really decided to test whether or not this sort of theory was right. And what he did was he took cowpox vesicles from the hand of one of the milkmaids, Sarah Nims was her name, and injected them into James Phipps. And Phipps was actually the healthy 8-year-old son of Jenner's longtime gardener. Jenner then waited 6 weeks, and then he took active smallpox from a patient's skin and injected it right into 8-year-old James. James didn't get sick. In fact, James Phipps never got sick, despite the fact that all the naysayers who didn't believe it injected James 20 times throughout his life with active smallpox to see if he would get ill. And to put the proverbial icing on the cake, sorry, for some reason, right? Jenner injected his own 11-month-old son, who also survived. So here's a fun fact. There was a cottage that actually Dr. Jenner left to James, and there's a plaque on it now that says, this cottage was left to James Phipps by Dr. Edward Jenner in recognition of his having been the first person that Dr. Jenner vaccinated against smallpox. And another fun fact is that it was Jenner who actually coined the term vaccination, from the Latin vaccinus, which means of or from the cow. So since 1796, we've had numerous vaccines, and they essentially all use the same method. They introduce live or attenuated, which means weakened virus, into people to provoke an immune response. And if you look here, you'll see measles, polio, rubella, cholera, yellow fever, influenza, meningococcal, typhoid, the list goes on and on and on. The point is that we have not, in 200 years, since 1796, about 2000, we didn't change how vaccine developmental technology worked. Even influenza virus that most healthcare workers in the USA and most people in Bermuda, I think, get, it still uses live or attenuated weakened viruses. In fact, the flu virus is grown in eggs. I don't know if you know that. And that's why they ask you, when you take your flu shot, have you been allergic to eggs? So it's even grown in eggs. Then by the early 2000s, scientists were openly asking, why haven't we changed this technology in 200 years? In fact, in Nature, which is the best scientific journal on the planet, as you know, in 2005, there was an article, a news article, that actually talked about an upcoming epidemic. And should we embrace a new vaccine technology to meet it? And look here, it says, pandemic fears hatch new methods in flu vaccine industry. Flu vaccine researchers are no longer placing all their eggs in one basket. Progress in vaccine technology has been slow because the vaccines do not provide enough economic incentive for manufacturers. So what was a researcher to do? There was no money in vaccines against infection. Well, go where the money is. Take the vaccines towards cancer research. And that's what exactly Igor Sahin and his wife, Özlem Tarice, they founded this company in 2004 that you've heard a lot about, BioNTech. And what did they do? They were making RNA vaccines against cancer. So just so you know, right around that time, as you might remember, we had two coronaviruses that just came up and showed their ugly head, SARS and MERS. Now, before we get into the vaccines per se, we've got to understand what the heck is messenger RNA. So I want to break here to give you a working scientific understanding, hopefully, of what this is. So to do that, I want to hark you back to your high school biology class and describe DNA, RNA, and protein as baking a cake. So DNA can be thought of as the ingredients, RNA is the recipe, and protein is the cake. One begets the other. The location of these three molecules in the cell is very important. Where are they located in the cell? This is what they look like. Where are they? The DNA is in the nucleus. That becomes important. The RNA and the protein are out in the cytoplasm. So nucleus and surrounded by cytoplasm. Okay. One more important thing to understand is that when we're talking about proteins, a lot of people think just about proteins that give you structure, your muscle, your fingernails, hair, etc. But protein is ubiquitous, and inside of our cells, proteins play a really important role, a vast array of roles, including they are the actual substances that our cells use if they're infected to signal our immune system that they're under attack by a foreign agent like a bacterial virus. They actually wave a flag outside of themselves, and it's a protein flag. So if our immune system senses a foreign protein flag, such as one belonging to a virus, it goes into attack mode. If they see it sort of hanging out there outside of a cell, it says, that cell's under attack, let me go help. So in essence, what Edward Jenner did was that he took the whole smallpox virus with all of its proteins, took the whole kit and caboodle, inoculated it into the blood of James Phipps, and that provoked a huge immune response. And believe it or not, that method still works today and still in use after 200 years. And in fact, for COVID-19, you actually can still make a virus that way. You can take the whole kit and caboodle, all the cake, and put it inoculated into the bloodstream so that all the viral proteins are actually into your bloodstream. And who would do that? Well, the Chinese did that. That's how they've done their vaccine. They've taken weakened coronavirus and done that. But in keeping with the times, the Novavax company here in Maryland decided, it is time to change. It is time after 200 years to change this technology. So what they said, instead of putting all those proteins in, let's break off one. Let's just put in the icing there. And part of the icing you've heard a lot about called the spike protein. So it's sticking up. Let's take that spike protein, that little bit of icing, and let's go ahead and let's put that and inject that directly into the bloodstream. And you know what? That works. Your immune system will respond to that. Now, Pfizer teamed up with Dr. Sahin and his wife and their company BioNTech, as you now know so well. And they said, well, let's use this messenger RNA to do this. And Moderna said, yeah, let's use the messenger RNA too. And the messenger RNA is injected into the cell. And what happens, it just has to go into the cytoplasm. And as soon as it goes in there, it's the recipe and it tells the cell to make its own cake. And it makes a cake that is identical to the foreign protein. You can also use DNA. You can actually use the DNA. And AstraZeneca and J&J, they actually use that. Now you're like, why is the DNA in that strange thing? Why? Well, because remember I said the DNA is in the nucleus. It has to go to the nucleus. So they had to come up with this vector, this chimpanzee adenovirus, to help guide that DNA all the way into the nucleus, drops it off, and then the DNA, once in the nucleus, can go ahead and make RNA and make the cake eventually for our cells. So how does this all work? So let's talk about now the mRNA vaccines. And let's talk mainly about Pfizer, because that's what everybody in Bermuda is getting. It's what I got. So the spike protein, and you can see my cursor here is here. And what Moderna did was they actually made messenger RNA for this protein, the instructions. They inject it in the cell, and once it's in the cell, it just has to go into the cytoplasm. It doesn't have to go into this white nucleus. They can just drop it off. And that's why they made this lipid nanoparticle you've heard a lot about. They can drop it off there. This thing dissolves, and very, very quickly, your own cells make the protein. It makes the spiral protein inside. As soon as the cell senses that there's a foreign protein in its midst, it pushes that thing out to the outside of the cell. And the cell is waving now its flag and saying, help, 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 I'm under attack. Now, you've heard a lot about antibodies, but that is just the lay press. There is something more important. And this is what I want you to take away from this lecture. That the thing that is most important, yeah, antibodies are important, but the T cells are important. You have two types of immune response, antibodies and T cells. And I can't tell you how important this is for T cells. Okay, what's the difference? The antibodies, they can be thought of as intercepting that virus from the time it hits your nose and as it circulates in your blood. Anytime before it infects the cell, almost like the Air Force, boom. Once it infects the cell, that virus infects the cell, antibodies can't see it. And your T cells take over. Your T cells deal with the virus after it infects the cell by destroying the whole cell with the virus inside. This is critical because I'm going to show you next when we talk about efficacy or effectiveness that vaccinated people, even if they do get COVID, they don't get severely ill. Why? Because the T cells have also been provoked by this vaccine response, and the T cells can destroy any infected cell before the virus can use the cell's machinery to grow and replicate itself. And it's probably the T cells why different people react to the virus once they're infected differently, and why some people get so sick and why some don't. It's all about your activity of your T cells. Okay, safety, and effectiveness. Now, safety, we sort of know about a little bit. I'll go into much more detail about safety. But efficacy or effectiveness has two definitions, very important. Now, you can either vaccinate against the disease or against the infection. Now, what's the difference? Well, the scientists at Pfizer at the beginning of the pandemic had to make a decision how to design the trials, but they have to do it quickly. You couldn't do both. So they made the no-brainer choice of vaccinate against whether or not this disease is going to put you in the hospital, make you sick, and die. Whether or not you can transmit the virus once you're vaccinated, we address that later. So, how did they do it? This is normally what's done. You have an exploratory phases when they were looking at cancer, and then you have a preclinical phase, and you could see these other 4 phases. Now, it's important to stress here, and I can't stress this enough, that each aspect of these phases, the most important thing is safety, 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 and then next, efficacy. So, first step, animal testing. You can see it says preclinical. With COVID, they used macaques. And they first looked at, was it safe? What side effects did the macaques have when they injected the vaccine into the macaques? Next, they said, okay, what kind of antibodies and T cells are being produced by the macaques? And then finally, just like Jenner did with James Phipps, they injected COVID to see what happened. And protection was strong. They did great. They said, okay, let's move on to humans. Phase 1. What's phase 1? 50 to 100 people. Safety, 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 safety. And we're going to use healthy people, college kids. Why? Because if anybody gets ill after being vaccinated, we want to make sure it's due to the vaccine and not to any disease. So we're going to have a young college kid who has no disease. We also in phase 1 want to know what's the highest and lowest doses we can use. What's that dose range? So we can have some idea of the dose to use of the vaccine. And we go to phase 2, 100 to about 500 people who reflect the demographics of the population, old, young, racially diverse. And we give that dose that we worked out in phase 1. Phase 3, you guessed it, more people in the Pfizer vaccine, 43,500 people about. And a mix of demographic people, including people who are sick. HIV people were involved, for example, people that were immunocompromised. Now, here is a sort of breakdown of who got the vaccine in their trial. 43,500 people. You can see half was in one arm, half was in the other arm, half got placebo in 4 different countries. The US had 77%, 50% were women. And you say, well, there weren't a lot of African Americans. Well, 10% African Americans, 2% multiracial. Usually that's Tiger Woods. He would put his name down as multiracial. So you sort of look, I would say 12% are minority, and maybe even Black. 13% Black in the United States, so that's pretty close. 21% over 65, 42% over 55 or over, and other disease are well represented, as you can see. All right. One of the unsettling ideas that happens all the time that people talk about is, how does a traditional process that takes 10 to 15 years happen in less than a year? The answer is the same as for most things in life. Human desire, especially if you incentivize this desire with what? $24 billion. The manufacturers had no risk. And the FDA regulators, they were allowed to streamline all red tape. The result was that the FDA was allowed to review all the human trials in real time, save in real time. So there was a rolling review. As soon as those clinical trials started, you can see they were rolling and reviewing, not waiting till the end. And the manufacturers, because they had no risk, they started at the end of phase 2 after they only had 500 people. Why? They had no risk. 24 billion worth of no risk. Okay. Vaccine effectiveness or efficacy. What is that all about? What was the result in terms of vaccine efficacy? So, sorry, I just had to take a little cup of tea. Whenever someone is arguing with you about science, just ask them to do one thing for you. Show me a picture, show me a graph, a table, something. Because this picture of the phase 3 clinical trials of the Pfizer vaccine is published in the New England Journal of Medicine, I'm going to show you in December 10th, 2020, is worth 1,000 words or more. So let's talk about 2 points in the interest of time. Not 1,000 words, but 2 points. First point, let's look at the bottom graph, not the inset. That blue line, it represents those patients who got the placebo, okay? Look at carefully. Each light-colored square actually represents a case of COVID. Now you see there are a couple squares in there that are all darkened. Those were severe cases of COVID. They went in the hospital. Now look on the red line. That's where people got the vaccine. And you can see that some people have circles. So they actually got COVID. And you'll see one that was colored in. And that represents a severe case. But I'll tell you, a week after this publication, the doctors realized that they actually had misclassified this patient. And they actually now, a week after that, so around the 17th, they said, there are no cases of severe COVID in the vaccine group. And you're going to see that over and over and over. So here, there was no vaccine group COVID cases. Okay, second point, look at the inset now. Look at that inset. It's a blow up of the first 20 days of the vaccine. Why? Well, a couple things to consider. First, look at from about day 0 to day 12. There is absolutely no difference between the vaccinated group and the placebo group. Why? Because it probably takes about 12 days after receiving your first shot for the vaccinated group to actually get an immune response. And then look at day 12, what happens? All of a sudden, the vaccinated curve, it goes flat. And it stays flat. Why is that important? Because on day 21, for those of you who have gotten Pfizer, you get your second dose. And look at what happened. The rate of infections doesn't go down. It stays flat. Interesting. Another way to think about this is that the first dose is like putting gas in your car. But then you put more gas in your car, and guess what? Your car doesn't go faster. So that's why you're hearing now so many experts arguing. It's all over any media that I look at as a doctor, arguing about when to have your second shot. Can they wait 6 weeks, like Dr. Jenner did with James Phipps? How long will the gas actually run the car just with that first dose? This picture now, just this picture, gives you some idea of that debate. And a lot of people are saying probably 6 weeks might be a good point to get the second dose. Now, scientific data loves to be validated. This is not the same graph. This is another graph. This is the Moderna messenger RNA vaccine that was published in New England Journal of Medicine on December 30th, 2020. It looks so similar. Why? Because both of these vaccines were essentially the same technology, and science is validated. And they are both 100% effective at protecting you from being hospitalized with COVID-19. The same thing. Now, safety. Talked about effectiveness, let's talk about safety. So, when they talk about vaccines, there are different grades, grade 3, grade 4, grade 3, and grade 5 is actually death. Grade 3 means some interference of your daily activity. Grade 4, you have to go to the ER or you got hospitalized. There were no grade 4 events. in the vaccinated group. 83% had site reactions, very mild. 2% had some grade 3 fatigue. They said, look, I had to spend that next day, but it was transient, it went away. They had to stay home the next day. 2% said, look, I had a headache, and they had to stay home. Now, this safety monitoring is continuing. And we'll talk a little bit about all the websites and so forth that are available and what's been gathered so far. A lot more information has been gathered since these trials, but they're basically showing exactly what the trials are showing. Heard about allergic reactions. Everybody's hearing about this thing called anaphylaxis and what's going on with anaphylaxis. First of all, it's very rare. It was said that it was 11.1 per million doses, you'll get it. 0.5 per 50,000 doses. That's been revised downward. Now they're at 1 per million. Why? Because the N value is gone up. More and more people are getting vaccinated in the world. So what we are telling people is that if you use an EpiPen and you're scared of being vaccinated, go to an allergist. There's an allergy clinic in Bermuda, and they actually can go ahead and look at the ingredients that are in the solution, the vehicle, and they can see if you're allergic to that, because you're not going to be allergic to the other stuff. You're not going to be allergic to the RNA, but you're going to be allergic to the vehicle. That's what people are allergic to. Test you against those ingredients. As of yesterday at 6 o'clock, and my father wanted me to share this with you, there are about 210 million people in the world who've gotten at least one shot. Israel's doing the best. Bermuda's getting up there. And this is February 9th data from Bermuda, because Bermuda reports, I think, every 10 to 15 days. So about 14% of Bermudians have been actually injected with at least one of the Pfizer doses. Now, if you don't remember anything else, because I'm at my 25 minutes, the theme of all these vaccines is 100% effective against severe disease. The Pfizer, Moderna, AstraZeneca, Johnson & Johnson, Novavax, the Sputnik, they're all 100% effective. And these are the ones that we have data from. Because it's the T cells that fight the disease. It's the T cells that determine how sick you will get. And it explains, as I said before, this idea of cross-reactivity, why maybe some people didn't get really sick and others did, because they had been exposed to something in the past, another coronavirus. Briefly, vaccine hesitancy, because we don't have time. There's a scientific objection versus myths. Now, when a scientist gets up there and says something, usually in this genre is designed to create scientific mistrust on questions that are impossible to answer, such as, how long will immunity last? Well, we don't know, because the first person that got it in the trial was got it August the 1st. So since August the 1st, you count the months. That's how long it lasts. Myths. All the myths that you will see and read about are usually rooted in one little small kernel of truth, and then it goes from there. Wait to take the vaccine. Vaccine causes infertility. Bill Gates tracking you, all that stuff. Let's just talk about waiting for the vaccine. I need to wait for months before I take this vaccine. Even healthcare workers are telling me this. So the researchers went back and they asked themselves a very simple question. Of all the vaccines out there, polio, rubella, typhoid, flu, measles, so forth, if there were any long-term side effects that occurred, what was the longest time interval from day one when the vaccine was given to when that long-term side effect appeared? The answer was 6 to 8 weeks. So can you get something after 6 to 8 weeks? In medicine, I've learned you never say never, you never say always, but I will say this, it is rare as rocking horse manure. I hope you understand my pun. As I'm being, okay, so you could also, what's happening is that they're monitoring this very, very carefully, more intensely than anything else with all of these different websites. And there's active monitoring. So I'm at my time limit. I gave you a brief history of vaccines. I gave you a little refresh on DNA, RNA, and protein. We talked about vaccine efficacy or effectiveness and safety. And finally, I hope I gave you some way of thinking about the myriad myths and objections that I think are floating around Bermuda and elsewhere. Thank you very much.